Okay, hi everyone. Welcome to this mini lecture on ethics. So we're essentially going to be discussing some of the ethical considerations you have to make in the context of assessment 2 for this module. So we've talked about how ethics is a significant component of your research proposal. So you need to think about whether your research is ethical and what considerations you need to make there. So what are the kind of things you need to be thinking about? How can you kind of check that your research is ethical? That's what we're going to be talking about here. One thing to bear in mind is there's no such thing as a perfectly ethical study. If, for example, you're doing studies with animal models, there could be some degree of stress and harm that is necessary to the animal models. But what you need to do is make it very clear you've considered this and you've gone to efforts to limit the amount of harm that's being done. And we can talk about exactly how you can do that. Similarly, with human subject research, there's always going to be some degree of chance that something could go wrong. So there's always some ethical issue at some point. But what you need to do is make it clear that you've considered this and that you've read the research around it and that you're comfortable that the research is ethically justified. Okay, so for example, in human subject research, you could say this drug hasn't been given to humans before. However, we have extensively tested this drug in mice and in non-human primates, and there's absolutely no negative consequences of treatments. There's no adverse events that have been recorded. It could also be a drug that has been tested in humans before and it's been shown to be safe and efficacious, but you want to test it in adolescents or in children, and the risk there is it hasn't been given to children before, and the way you discuss this risk and mitigate this risk, the risk of anything going wrong, is you say it's been extensively tested in animal models, it's been shown to be completely safe, it's been extensively tested in human subjects that are adults and it's been shown to be safe, again extensively. So now we're looking at this small scale study in children. We're going to use a minimal dose. We're going to monitor everything very, very carefully. So again, you're showing that you've thought about the ethics and you're taking these practical steps to mitigate this risk of things going wrong. Again, it applies to animal models too. So one thing we talked about, and we'll talk about it a little bit more in this session, is the idea if you're doing, say, research with mice and it's going to cause some degree of harm and stress to the mice, and you are proposing to do the research using 20 mice, is it possible to do that research in 10 mice instead? Can you still produce useful results? Are there other studies that are using 10 mice and doing something similar? If so, then it's unethical to use 20 mice. And even if you can't come up with exact numbers, you can say you'll be using the minimum number possible. And this study, and you reference it, has used this number of mice. So we're going to be using that number of mice because it's a new drug. So we don't know the exact number. We'll need to test it thoroughly, but we're going to follow the kind of protocol that's been established in this other study. Because then you're showing you're thinking about ethics and you're showing you're making these ethical considerations. We'll also talk about research in a lab-based environment that is in vitro, so doing tissue culture experiments, so working with cells. And the question here is, how could this research possibly be unethical? Right, it's just cells, who cares if we harm them? We thaw them out the freezer, we can grow an infinite number of them, so who cares? And the answer is, in that context, we're generally thinking about the ethical considerations to be made with regards to the researcher. So if you're investigating HIV, which is one of the potential topics here, is it being done in a safe environment? Are you doing it in a lab environment that's appropriate for HIV? So in the UK, you need a category 3 laboratory to do experiments with HIV. So you need to state that it will be in a category 3 laboratory. You need to state that there's going to be protocols in place in case something goes wrong. These are things that you again need to discuss. Okay, and these are the things we'll be talking about as we move through the lecture. So we have a slide here, and this slide is based upon the idea that when we are doing research, we have to think about the participants in the research and how we can protect them. And the ethical considerations we have to make are very, very high for human subject research. So this is on the right hand side of the scale, and they're much lower if we're working in cell lines and we're working in primary cells. And we even talked about the idea that if we're working in cell lines, so just doing tissue culture in the lab, we really don't care at all about the well-being of the cells. It doesn't make sense to. So our main ethical considerations become concerned about the experimenter, the person who's doing the experiment, because there's basically no ethical considerations to working with cells. The only thing you might want to consider is gain of function. So if you're doing experiments with viruses, you need to make sure you're not doing experiments where you're making the virus stronger and better at infecting and stuff like that. I don't predict any of you will be proposing this kind of project, but it's something you might see in the literature. They call it gain of function experiments, and they're ethically complicated. But again, most of the experiments and projects you'll be proposing won't be anything like this. It will be maybe drug screens or looking at mechanistic interactions between proteins within cells. So most of the ethical considerations here will be towards the experimenter and just making sure the experimenter is safe. If we move from cell lines to primary cells, we do have some additional ethical considerations to be made here. And that's because primary cells come from individuals. So we have a finite supply. Okay, so the barrier for our research, the justification for our research gets a little bit higher. We can't be completely just messing around, trying random things out, because it's unethical, because there's a cost there. We've taken the cells from an individual, we've taken their blood, or we've taken a biopsy. So someone has suffered a little bit, right? That's practical suffering. So we have to explain why our research is important enough to justify that small amount of suffering. So there's a little bit of an ethical consideration there. If we move into animal models, the ethical considerations become even more significant. You won't see experiments in mice or rabbits where the experimenters are just messing around doing weird and random things, because they are making the animals suffer and there's no justification for it. You know, they don't have any firm reason to believe they're going to get important expected outcomes. So usually when we're doing research in animals, that research is backed up, it's supported by research that has already been done in cell lines. And that's why we have the workflow that we talk about, where you identify useful drugs in cell lines and primary cells, and then you test them in animal models. Because once you have the first experiments, you have the justification to conduct the experiments in animal models, because you do have this reasonable expectation that the drug might work, and you might then be able to progress it further. Very often, if we're doing experiments like this in complex animal models, so non-human primates in particular, we will have not just done preliminary work in cell lines, we'll have also done it in less complex animal models, because again, it's this workflow. You go step, 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 because you need justification to do research in an animal as complex as a rhesus macaque monkey or a chimpanzee. And some people will say, you know, research in cell lines isn't enough to justify this. You need to show the drug works in mice or in zebrafish before you jump to using these complex, intelligent animals. And that's generally the ethical approach we take. And remember, this is a scale of animal models where the more complex the animal model is, the higher the ethical considerations are. Just remember, it's intuitively clear that killing a bug, killing a butterfly, you know, it's not a nice thing to do, but it's very different to killing a chimpanzee, right? We kind of accept that intuitively, and it's built into the ethical codes, it's built into the guides, and these formal guides tend to classify these animal species based upon kind of neurological complexity. They have these criteria. Can they feel pain? Can they respond to pain? These types of criteria. So just think about this. If your preliminary research, so the data you found through PubMed, shows that the drug you're interested in works really well in cell lines and or primary cells, but you haven't got any animal experimentation published, then it might be a better idea to try it out in mice or zebrafish rather than trying it out straight away in rhesus macaques, so these complex non-human primates. Also remember that for different diseases we have different animal models, so it's not just a case of randomly picking an animal model that you think is suitable and should be the next step. You need to check, do we have a mouse model for HIV? Do we have a chimpanzee model for SLE? You know, many pathologies we don't have a huge number of animal models, so you have to make this consideration. And if you are proposing we do research in rhesus macaques, so these non-human primates, even though we don't have any other animal model data, and the reason is rhesus macaques are the only animal model we have for this disease, that's fine, but you need to explain it. Okay, there's other ways that we can make animal model research as ethical as possible, and we'll talk about those as well in a few slides. Once we get to human subject research, this is the research that has the highest ethical boundary, so it has the most significant ethical considerations, because again we intuitively understand that causing stress and harm to a human is very, very different to causing stress and harm to an animal. So we have to be very, very careful and considerate and cautious when we're doing human subject research, and we need to justify this research extensively. We absolutely need to have preliminary data in some animal models. Usually 2 animal models is the minimum, but for some pathologies you will see human subject research being done even when there's only 1 animal model that's being used, and this is almost always because only 1 animal model for the disease exists. And in those cases, it's very clearly explained that this is the situation. Okay, so think about this. If you don't have animal model research in the literature that supports the drug you want to move forward with, you can't propose human subject research. It's not ethical. If there's data showing the drug is safe and efficacious in mice, but there's no data showing it's safe and efficacious in non-human primates, and you know there is a non-human primate model for the disease, then again you can't propose human subject research. You should be proposing research be done in these non-human primates. Think about this workflow, think about this process, this pipeline, and why it exists. It's partly practical, just because we don't want to spend huge amounts of time and resources testing random drugs in chimpanzees in these really, really expensive, complex experiments when we don't even know if they work in cells yet, so we have no evidence they work at all. That's one of the considerations. And the other consideration is, again, as we've talked about here, ethics. We need to think about both these things. Okay. So just some more information on these cell lines. So what we mean by cell lines, these are immortalized cells, so they're cancer cells that have been obtained from individuals historically, and we essentially freeze them and we can thaw them and make basically an infinite number of copies of these, because they grow and grow and replicate, because remember, they're derived from cancer cell lines, they're immortalized. So in terms of ethical considerations, they're really minimal in terms of harm to the cell lines. We don't care about that. The things we do need to think about is whether or not our experimental approach is safe for the experimenter, the scientist doing the work, right? But in terms of the model, we really don't care. Beyond ethics, you do need to think about the usefulness of your model, and we talked about this in the lecture, but I'll touch on it here. Essentially, these cell lines have low physiological relevance. So the experiments we're doing in these cell lines are not going to represent what we would expect to see in an actual human, because these are cancer cells. They're being grown in a lab on a plastic plate. So the situation is so different to actual disease. And again, this is another reason why we use this pipeline, because as we go from cell lines to primary cells and animal models, the model we're using is more and more similar to what we see in human infection or human disease. So the data we get is more and more useful. So it's this step-by-step process. We're checking if it's safe, and we're checking if it's efficacious. So we're checking if it works. And each step along the way, we're getting rid of any drugs or therapies that don't look like they work. And we're moving forward with the ones that look like they do. And as we get from model to model, so we increase in complexity, the data we see is expected to more closely represent the human physiology, so the actual disease state in humans. And if we think about primary cells, we already see that. So primary cells are obtained from an individual. These aren't cancer cells necessarily. I mean, you can get primary cancer cells, but that's not what defines them. What defines them is the fact they've been obtained from an individual. So they represent actual human physiology better than the immortalized cell lines, because immortalized cell lines have been grown for decades and decades in the lab. Primary cells are directly obtained from an individual. Again, remember, these primary cells being obtained in this way means there's a cost there, because we're causing some amount of stress and harm to an individual. And we have to consider that. We have to be a little bit more careful, a little bit more considered in how we use them. And that's why a lot of the time we will test the drugs in cell lines and then we'll move to primary cells. Remember, you have to think about the primary cells you're using. So if your focus is, again, HIV is one of the diseases. So if your focus is HIV, you're not going to take primary epithelial cells from an individual, because HIV doesn't infect epithelial cells. You're going to take primary CD4 T cells and macrophages, because those are the cells that are infected in vivo. So if you are using primary cells, look at the literature, see what types of primary cells are being used. We've talked extensively about this idea of animal models and the ethical considerations here. They vary. The more complex the animal species, the higher the ethical considerations are, because again, we just accept that the more complex an animal is, the more significant it is to cause harm and stress to that animal. You might disagree with me there. It's absolutely fine. You have the right to, but this is the opinion of the scientific community rather than my own. And again, this is reflected in the ethics guidelines within and outside institutions. Okay, so for the purposes of how we discuss ethics here, we'll have to kind of follow this approach. We can think about the animal models as scaling all the way from Drosophila, so fruit flies, to zebrafish, to mice, to rabbits, and then all the way up to the non-human primates. And we've talked about the types of considerations that we have to make in terms of picking an animal model. In the few slides after this, we'll talk in more detail about how, once we've started to focus on an animal model, we can look at our research approach again and see how we can change it to make it even more ethical. And finally, we have human subject research. So as we said, the most significant ethical considerations are here. If we are proposing human subject research, we need to have all this background data showing that it's safe and efficacious. You also need to look in the literature at other clinical trials and see all the processes put in place to make sure the participants are safe. So read in real detail the other clinical trials that are done, particularly in the context of your chosen disease. Look at how they monitor individuals. And again, feed these ideas into your research proposal. And when you're thinking about these things like monitoring the human subjects, again, look at the literature. In clinical trials, they'll check on the participants at regular intervals. And what are they checking? Are they checking blood pressure? Are they looking for specific side effects? Are they looking at specific biomarkers or components in the blood? Again, it will vary dependent upon the study you're doing, and you need to look in the literature at other clinical trials that have looked at treatments, so prevention or treatment in the context of your specific disease, and you should follow their guide and you should reference them. Okay, so there's this concept of reduce, refine, replace. So these are the 3 R's of animal research. So these are 3 principles that we have to follow to make sure our animal model research is ethical. So we're in a situation where we've decided on the research we want to do. And now we're kind of thinking about making it as ethical as possible. Okay, so the first thing is we can reduce the number of animal models that we're using. And we talked about this concept earlier. If we're using mice and we're thinking about using 20 mice, can we use 10 instead? Can we use 6? What do other studies use? So think about the minimum that you can use based upon the literature and go with that number. Okay. Also think that there might be dropouts and things like that, but other studies will account for this. You know, there could be mice randomly dying, but again, other studies will account for this. See what they've done and then kind of use that as a baseline to help you make a decision here. This isn't something that we're going to be super exact about in the case of, oh, they've proposed 8 instead of 6. That's a big problem. That's not what we're doing here. We just want to encourage you to at least think about this. Say these other studies have used roughly this number, so between 12 and 20. So we're going to go right in the middle of that range based upon them showing that you can do a nice 6-month study with mice and get usable data from this number of mice. What we don't want is you to just pick 100 or pick 50 just because it's a nice round number and you want to get lots of data. Okay, that's not the approach when we're doing animal model studies. We want to balance getting enough data with minimizing the number of animal participants. So we just want to show that you've thought about that here. In terms of refining, so we need to refine our specific approach to the study to make sure we're doing things as ethically as possible. Again, look into the literature at how these animal models are treated. If you'll pick a specific disease and then you look at the other papers that have used the relevant animal model for this disease. Look at the considerations these have made. You can again use them as a guide. We're not asking you to kind of reinvent the wheel here. You need to look into the literature and see what other studies have done. That's absolutely fine. We encourage it, and you just need to reference those studies. So if you have to euthanize the animal models at the end of the study, you say you'll be using the euthanization protocol that this other study and this other study has done, because they have shown it's the most ethical way to euthanize the animals. Okay. This idea of replacement as well falls right into that concept of the scaling ethical considerations that we talked about. So remember, the more complex an animal is, the more significant the ethical considerations are. So we can improve our study, make it more ethical by using a less complex animal model. So if you can replace your animal model with a less complex animal model and still get useful data, or still predict you will get useful data, then you should do that. So again, if you find yourself proposing a study with non-human primates, but there's no research into this drug in a rabbit model for your disease or a mouse model, and you know those models exist, you should seriously think about replacing the model you're using with the more simplistic model. Okay. Again, if you're trying to do animal model research and there's not sufficient data from the cell lines or the primary cells, there's some unanswered questions, or the data that does exist is kind of weak or weird, you don't like it, there's flaws in the research, then there's nothing wrong with you proposing rigorous in vitro research, so using cell lines or primary cells, that is essentially taking a better methodology than the existing studies. So you're improving upon the studies that have already been done in some very specific ways. That's absolutely fine as well. And you can say one of your considerations is ethics. You can say the research in cell lines and primary cells is not strong enough to justify us using this mouse model for the disease. So considering these ethical concerns, we believe we should do an additional extensive rigorous study of this drug in this cell line or in these primary cells, and this is exactly how we'll do it. Okay, so the main thing here is that you're thinking about the ethical considerations. You're showing that you've thought about these things. You haven't just picked an animal model at random. You haven't just picked a random number of animal participants to do here, and you haven't not thought about the methodology at all. And again, all your information for this is going to come from the literature. Because it's level 6, you need to be jumping into the literature, looking at other studies that have done similar things and referring to these studies. So thank you for listening. This should have been really useful in helping you make these ethical considerations for assessment 2. So in summary, just make sure you're thinking about this ethical component. Make sure you can clearly see in your submission where you've made specific points about ethics. There's no such thing as a perfect study in regards to anything, but here in regards to ethics, we just want to see that you've considered these ethical points and you've made an effort to ensure your study is ethical. And again, this should be supported by the literature, and you should be referencing the literature to show where you've made these specific decisions and adjustments to ensure that your study is as ethical as it can be. Okay, thank you.